Of trying to run this entire. Awesome. Okay, this is the first lecture in the second year of running this Symbiogenesis initiative. The goal of the initiative is to bring together people thinking about how endosymbiosis and symbiosis could be used to engineer biology. Noa Sher is the CSO at Minovia Therapeutics, which is a cell therapy or mitochondrial augmentation therapy company, and also like a personal inspiration of mine when I think about my career. Her team leads the company's mitochondrial augmentation technology platform, and they're already like a clinical stage company. So this is a really great opportunity for anyone thinking about how to translate their innovation in a way that actually impacts the lives of patients and the world around us. And with that, thank you very much, Noah, and you're welcome to start.
Happily, Liam. It's really a pleasure to speak with you all. You can see the slides. Okay.
Yes.
Yes, we can see.
Okay, perfect. So I debated kind of as I was building this deck how to present it to you guys because I'm assuming it's a very scientific audience. And so I'm gonna go deep into the science and the preclinical stuff on some place, but I think I want to just tell you the story of how we kind of balance between all of the preclinical studies and the clinical studies and how you actually take an idea from the concept, the first thing that you thought about, before you went into the lab and all the way into the clinic and some of the challenges that you run into the way. So I hope it'll be interesting. Feel free to ask questions anytime along the way because I love interaction. So, don't hesitate or wait to the end. So Minovia was founded on with the goal of using mitochondria as a therapy. And what we developed over the first few years of the company was what we now call mitochondrial augmentation technology. And it's basically founded on the principle that isolated mitochondria can go into cells, so re-enter cells, just like what happened billions of years ago when the first endosymbiont went into cells. There was a publication in the 1980s that showed for the first time that isolated mitochondria can enter cells. It was later shown also in the 80s that they can even, under the proper selective conditions, rebuild the normal mitochondrial content of cells, so actually take over the cells and restore functionality. And then there's also a lot of studies showing that mitochondria can transfer from cells to cells. This was shown extensively in vitro and more recently in vivo. And there's always a question, and this is still ongoing debate in the field of whether this is physiologically relevant. Does mitochondrial transfer occur at physiologically relevant levels? And I think there's more and more data out there suggesting that it does. So we took this kind of concept and developed it into a process. And actually, we found a way to isolate and cryopreserve functional mitochondria. And that piece was really important because if you can't cryopreserve, then it's really hard to make something into a product. So you can do a quick and dirty prep of mitochondria and immediately put it in a dish with cells, but really making it into a drug product needs that cryopreservation step. And what we did at first was demonstrate that we can isolate mitochondria and then incubate them under the proper conditions with human hematopoietic stem and progenitor cells. And you're seeing these here. So the green is isolated mitochondria, the red are the endogenous mitochondria, and you can see that they've entered the cells. And we've since done this with many different cell types, so LCL type cells and CAR T and so forth, MSCs. And really, we need to find the precise conditions to make this work to extents that are reasonable for each cell type. Why do we start with hematopoietic stem cells? They build the entire hematopoietic content of the body, so immune cells and all of these tissues. But really, we know that from many studies that if you have mitochondrial dysfunction in hematopoietic stem cells, you get cytopenias. So you get anemia, thrombocytopenia, and so forth. You get immune system dysfunction. You have a loss of the hematopoietic stem cell reservoir. But also you get many other dysfunctions. So it's not just the hematopoietic system, but because that system just flows into every cell and tissue in the body, you get crashes in multiple systems. So there have been a bunch of really recent works showing that if you have mitochondrial dysfunction, even in single cell types, so just in T cells or just in myeloid cells, you get aging, so you get brain phenotypes and you get kidney phenotypes and liver phenotypes and cardiovascular phenotypes and so forth. So there really is this connection between hematopoietic stem cell well-being and hematopoietic well-being, and then organismal well-being. So that was kind of the very high level concept. We're going to try to treat hematopoietic stem cell mitochondrial dysfunction by bringing in healthy functional mitochondria. The first couple of years of the company, we did studies with any cell we could get our hands on, so fibroblasts and what have you. Um, but then that kind of funneled into how do you make that into a product? And what we decided to do was test mitochondrial augmentation technology first on Pearson syndrome patients. So let me stop for a second with the MAT and tell you what Pearson syndrome is. Pearson syndrome is a really deadly disease. It's a pediatric disease that starts with bone marrow failure, and it's caused by a mitochondrial deletion. So, as a reminder, the mitochondria have their own genome. It's about 17 kb, so 17,000 base pairs. And in Pearson syndrome, anything between 1 and 10 kilobases is just chopped out of the genome. What that looks like on a Kaplan-Meier is this. So about half of the patients die around by age 5, and then the rest succumb to the disease before their 20th birthday. And on the right, you could see that it's really a multisystemic phenotype. So all of the patients start with anemia, that's these red blocks. And then as they grow, those patients that are able to survive the first couple of years of anemia start to develop additional organ system involvement. So all of these different colors are different organs that are involved, the cardiac and the muscle and the nervous system and the liver and the kidney and so forth. And the variability in different patients is not entirely understood. It might be because of the level of heteroplasmy in the different tissues. So just as a reminder, each cell in the body has hundreds or thousands of mitochondria, and each mitochondria has 2 to 10 mitochondrial DNA copies. So heteroplasmy is the level of dysfunctional versus functional mitochondrial. Suffice to say, it's a horrible disease, and we decided we're going to try to treat this disease. There is no, or there was no animal model for Pearson at the time. So what do you do if you want to treat patients and there's no animal model to study? What we did is we did what we could with cells. So to prove in vitro that the technology works, that we can bring in mitochondria to cells. And we do a toxicology studies. So you just want to make sure that you're safe. And then we came up with an idea of how to treat these patients with minimum risk to the patients. So the technology in the first generation treatment was as follows. You harvest their hematopoietic stem and progenitor cells by mobilizing their bone marrow to the peripheral blood and isolating CD34 cells. Those are the hematopoietic stem cells in humans on a CD34 column. We previously harvested mitochondria from the mothers. And why from a maternal blood unit? Because we said, I should have mentioned, Pearson syndrome is a de novo disease. So the patients should have just gotten normal mitochondria from their mothers, but they did not. And so the best place to take mitochondria for these patients was the mitochondria from the mother. So we took a blood unit from the mother, and that was the mitochondria we used to do mitochondrial augmentation on the hematopoietic stem cells. We qualify them to make sure that the mitochondria have indeed gone in, and of course, and then we use into the patients. Any of you who know about hematopoietic stem cell therapies, most likely conditioning is done to make room in the bone marrow niche for these patients. And what we decided to do is not to do conditioning. Firstly, because Pearson syndrome is associated with acellularity. And secondly, because mitochondria are really susceptible to all of the conditioning reagents. And we said, first, do no harm. So we didn't want to harm these patients in any way. And we started to treat patients. So the first few patients were treated under compassionate use protocols. What you see here is the renal function in the first Pearson syndrome patient that we treated. So you can see the creatinine before treatment was rising and the glomerular filtration rate was decreasing. And both of these stabilized after the treatment. So this was actually a patient who was already on the way to getting a kidney transplant, and that stopped. And we were able to postpone that kidney transplant for 5 years after treatment. This same patient had been not gaining any height or weight. So what you're seeing here is a height SDS, so it's a standard deviation score, how far the patient is from the normal for that age group. And he was just getting further and further away from height and weight. And after treatment, he started to improve in both height and weight. The second patient that we treated was actually the first patient that we had a bone marrow biopsy for. And you could see that her bone marrow cellularity was 20 to 40% before treatment and 40 to 70% after treatment. And then we actually treated another compassionate use patient with a different mitochondrial DNA deletion. So this is a deletion that doesn't, it's not systemic. You don't see in all of the organs, but mainly in the brain. And she was having daily epileptic seizures prior to treatment. And you can see that these normalized after treatment. So the spike index went down and down until it was really zero after treatment. So we started to get really excited by these results, and we said, okay, we're going to stop with the compassionate use and go into a clinical trial. But in parallel to setting up that clinical trial, we're trying to understand how we impact all of these different organs. So how do you come with a treatment on hematopoietic stem cells and impact the kidney and the brain and all sorts of other organs? So we did a few preclinical studies, whatever we could at the time. And it's important to say that we can't test any of these things on healthy CD34 cells because there's no dysfunction there. So if you're trying to assess what the mechanism of a treatment is, you need to work with dysfunctional cells. So one of the models we were using, again, because there is no Pearson syndrome in vivo model, was the Paul G mouse. It's a general primary mitochondrial disease, but it's nuclear encoded. And we treated the Paul G mice with augmented cells from a mouse that was genetically modified so that the nucleus is red, sorry, and the mitochondria are green, so that we can trace to see whether mitochondrial transfer is occurring. And what you would expect to see there in the hematopoietic tissues that we were looking at are the original Paul G cells, which are going to be not a red nucleus, not green mitochondria. The transplanted infused cells that had undergone mitochondrial augmentation are going to be red and green. And any in vivo transfer is going to be green mitochondria into a non-red nucleus because they've transferred from these infused cells to the Paul G cell. And what you can see really nicely is that mitochondria were transferred in the hematopoietic systems in the blood and in the bone marrow, and that rises over time. And we do see continual mitochondrial transfer. And we believe that this is a continual process that's happening in each and every one of us. So at least in the hematopoietic system, we think there's constant exchange between different cell types. And this has been published, so you can look at that more deeply. We also had the rare situation where we were able to access cells from a Pearson syndrome patient because unfortunately, this patient passed away before we were able to treat her, and the parents were kind enough to give us her cells. So we could augment those cells with blood-derived mitochondria and treat a immunocompromised mouse model. And then we looked after several months to see what happened with that mouse's hematopoietic system. And you can see that MAT improved engraftment of the augmented cells and also had rescued immune cell production, specifically T cell production in this case. So we started to see the effects of mitochondrial augmentation on the immune cell system. We also did some studies using aging mice and saw improved kidney function and improved neurocognitive function as well. And at this stage, our working hypothesis was one where we bring in functional mitochondria into hematopoietic stem cells. They home to the bone marrow. They improve, this I'll show you a little bit later on, they improve proliferation into the various hematopoietic lineages, and this changes the different non-hematopoietic tissue function. I will say at this stage, what we thought was happening, and this is no longer the case, I'll get to that later, but it's very confusing to people who were with us since the beginning. We thought this was like a gene therapy-like approach, so that we're bringing in exogenous mitochondria, and that those exogenous mitochondria are going to proliferate and slowly take over so that you'll see a reduction in heteroplasmy in the hematopoietic tissues, but also in other tissues due to mitochondrial transfer. We've later moved away from that mechanism of action, which I'll show you a little bit later on. So we proceeded into a clinical trial. This was very successful. We had 5 patients treated. They all rose in what was then our primary endpoint, which was the International Pediatric Mitochondrial Disease Score. Actually, the 4 Pearson syndrome patients improved in that at the 6 and 12 months time point. The one patient where we didn't really see a change was a patient who was actually a KSS patient, and she had a very almost maximum score to begin with. What's interesting when you're doing clinical trials is that it's struggling to find an endpoint that is both clinically objective and clinically meaningful. And those are both 2 criteria which the regulatory, so the FDA find critical. And I showed you the slide earlier with the variability between Pearson syndrome patients. The reason we like the IPMDS scores is that they have questions on each of the different organ systems. So that can really take a heterogeneous population and summarize improvement in all of them. But the FDA didn't like that, so we went and we looked for a different endpoint to do on this patient population. We actually funded a natural history study at the CHOP. I won't get too much into this, but what they did show was that there was a continual decline in the height SDS of these patients. So how far they are from the height of the age-matched children. And you can see these steep drops. So this is a patient that goes from being 3 standard deviations to 4 to 5 to 6 over the course of maybe a year. And Pearson KSS patients also have a severe drop. And now this is the endpoint that we're using in our clinical study. And what's important to note is that height SDS, the height is not so much the issue, right? I don't think anybody cares in the Pearson syndrome patients if they're a few centimeters higher or lower. But what we think, and we're actually doing a study to prove now, is that the height SDS reductions are indicative of the organ dysfunction and the various organ dysfunctions. So if you have anemia, you're not going to be able to grow. If you have GI issues and you're not able to uptake food, you're not going to grow. If you have pancreatic issues or appetite issues or wakefulness issues, these are all kind of conglomerated into this height SDS parameter. What we did do is go back to the clinical study data and look to see whether we saw changes versus the natural history. So if you look at the natural history data that I just showed you and say, how much does a patient get further and further from the norm without treatment, they drop about a half a notch every year. So you're at minus, you lose one height SDS after half a year, another one after a year, and so forth. And after treatment, that's the blue bars, we seem to stabilize for at least 2 years. And you can see this on the right by a patient by patient. So when you look at the patient as his own control, in the 6 months before treatment versus the 6 months after treatment, or the 1 year before treatment versus the 1 year after treatment, the patients are doing much better after MAT. We also always ask ourselves about durability of effect. So this is all from a single treatment of mitochondrial augmentation technology. You could see here at 2 years, 3 of the patients were still improved, whereas 1 had already lost the improvement, so would need redosing. So all that was kind of the first couple of years of Minovia with Pearson syndrome patients. And then you have practicalities. So one of the things that we ran into was that this was a really personalized medicine. We were making mitochondria for each patient from his own mother, which is really challenging to do on an industrial scale, especially as we proceeded with more and more discussions with the FDA, and they were asking for more and more quality control tests in the mitochondria. And from a single blood unit from a mother, you only get so much mitochondria. So it was really challenging to do. And we also wanted to expand into additional indications because, as you may imagine, Pearson syndrome, I didn't say, is an ultra rare disease. There's 100 patients in the world, maybe 100, maybe 400, but it's definitely on that order of magnitude. That's not a huge market. So we wanted to, in addition to treating Pearson syndrome patients, expand into other places. And so what we did was we transitioned from harvesting mitochondria from the mother to an allogeneic mitochondrial product. And what we chose as our organ from which to harvest mitochondria was donor term placenta. So these are placenta coming from C-sections. They're scalable. So from a single placenta, you can get hundreds of vials of mitochondria. And then you do all of the quality control tests on a couple of vials, and you've got many more left over for treatment. It enables us to treat additional diseases, so diseases that are inherited and therefore it doesn't make any sense to take from the mother, or diseases of elderly people where the mother would no longer be relevant. And the placenta has many, many very functional mitochondria. So it's really like the best source of mitochondria. It's an ephemeral tissue. It's thrown away at 9 months. So you're never going to get a younger tissue than that from which to harvest mitochondria. And the other bit that was important from a mechanistic point of view was that you can track the persistence of these mitochondria now in the clinic. So when we're choosing between donors and recipients, there was always the question of, do you need to match mitochondria? And I'll get to that in a few slides. But there was also an issue of how long does the mitochondria persist? when you treat. And once you have differences between the donor and the recipient mitochondria, we can use single nucleotide polymorphisms to differentiate between those and then track them over time, either in vivo studies or in the clinic. So we transitioned from the first generation product to the second generation product. We reassessed all of the methodologies we were using. We reassessed all of the the entire process. And we did a couple of single cell studies with Leif Ludwig and Caleb Leroux to see what is the extent of exogenous mitochondria going into the cells. So we used a mitochondria attack-seq that they developed when they were at the Broad to look at samples that were augmented. And what you can see here is that anywhere between, depending on the experiment, 30 and 50% of the cells were augmented. And the level of augmentation was up to 30% exogenous mitochondrial DNA. So some cells are receiving very few mitochondria, 2% exogenous, 4% exogenous, and other cells are receiving high levels of exogenous mitochondrial DNA. Um, and another important thing that we did in this experiment was to look at which cell types within the CD34 population are being augmented because we wanted to make sure that the true hematopoietic stem cells and not just the progenitors are augmented, because that would give us a longer durability of effect. And you can see that these hematopoietic stem cells, as well as all of the progenitors, are being augmented. So what does the second generation product look like? It looks exactly like it did in the first generation, except for now we're augmenting with placenta-derived mitochondria. We treated Pearson syndrome patients. This is an ongoing clinical trial, and we want to see that we're getting the same effect as we got in the first generation product. And here, what you could see is the height SDS. These are preliminary results from the trial, but you could see, for example, in this patient that prior to treatment, they were experiencing that drop that we saw from the natural history, whereas after treatment, he seems to be stabilizing so no longer losing height SDS. How do we expand beyond Pearson syndrome? So we were trying to think, what is the best disease where we think we have the best chance of success? And one of the things that's common in Pearson syndrome is a phenotype known as sideroblastic anemia. The bone marrow dysfunction has ring sideroblasts because of mitochondrial iron deposition. And another disease that has this sideroblastic anemia is low risk MDS. And what was actually interesting is that if you look at Pearson syndrome patients, about 10% of them actually have leukemias, including most of them have MDS. And that is extremely rare in pediatric patients. So we started to suspect that there's a link between mitochondrial dysfunction and causation of MDS. And so we did a clinical trial it's called a non-investigational drug clinical trial, just to access patient samples. And we started to look at different mitochondrial biomarkers that we've been developing. So we look at the heteroplasmy, so the number of mutations in the mitochondrial DNA. And it's known that in a normal population, this is work from Vamsi Mutha's lab, there is a rise in heteroplasmy over time. So we're all going to amass mitochondrial DNA mutations over time, unfortunately. But in these MDS patients, those levels were off the charts relative to the age-matched controls. We also looked at GDF15, which is a biomarker of mitochondrial dysfunction and actually of just general stress. And you can see that the MDS patients, for the most part, have a much higher level than the age-matched controls. And then we looked at two different biomarkers that we've been developing. So mitochondrial score and glycolytic score, which kind of look at the balance between mitochondrial function and glycolysis. And you can definitely see that the controls and the MDS patients differentiate between themselves. So there's something really off with the mitochondrial functionality of hematopoietic cells in these patients. And that kind of persuaded us to try to go to do some preclinical work in this patient population. We developed two methods. So one of them is an in vitro erythroid differentiation method based on literature. It's a 2 to 3 week assay where you take hematopoietic stem cells and you let them differentiate into the erythroid cells. And the reason for this is because MDS is primarily a disease associated with anemia. And we also did an in vivo bone marrow reconstitution assay, so looking whether hematopoietic stem cells can reconstitute into an immunocompromised mouse model. And as you can see in both cases, healthy cells work fine. MDS patient-derived cells are horrible at either differentiating into erythroid cells or reconstituting the bone marrow. And what happens after MAT is that you get a much higher level of erythroid cells, both mature erythrocytes and terminal erythrocytes, which are really where you want to take your hematopoietic stem cells, right? You want to take them out of anemia for this patient population. Um, when we started to work on MDS cells, we knew that there was a mutation called SF31B that is associated with mitochondrial dysfunction. And our initial thoughts were to take a clinical trial in that direction. But what we actually saw when we started to do these studies was that almost all of the cells, at least it was more than 50% of the cells were responders in this assay. So it seemed that it didn't necessarily associate with that particular genetic mutation. So a high proportion of patient-derived MDS samples were able to get benefit from MAT. And this was actually true not only in the low risk samples, but you were seeing higher proliferation in the intermediate risk and in the higher risk samples as well. Another important point here is that there was no correlation between the haplogroup matching and efficacy. And that was important for us in thinking of whether we want to do haplogroup matching in the first ever trial of MAT using allogeneic mitochondria. So once we saw these exciting results from the in vitro, we moved into the in vivo study and asked whether we can rescue this phenotype of MDS cells not doing any bone marrow reconstitution. And here you see one example where you could see that there was a much higher level of reconstitution of an MDS sample after MAT in both the bone marrow, the spleen, the peripheral blood, and we were actually able to see mature erythrocytes in the bone marrow. This result was replicated in roughly 50% of the cells. So 50% of the patient samples were responders. And again, we didn't see any correlation between haplogroup matching and efficacy. And eventually we decided that there is no need to match haplogroups in a clinical trial. What was also important for us from a clinical perspective was that we do no harm. So myelodysplastic syndrome is associated with a progression to AML, to acute myeloid leukemia. And one of the concerns here, despite the fact that we're not doing anything that's directly manipulating the genome, was that we didn't want to, in some way, cause a more rapid progression of the patients from MDS to AML. So in all of these in vivo studies, we took samples before and after. So the in vivo studies ran for 4 months, and we saw that there was no change in the level of mutations, and there were no rise in the AML-associated mutations. So we felt fairly confident that we're not going to do any harm to these patients. And we used a variety of different methodology. I think one of the important things that I always stress is that in the mitochondrial transfer field and in the mitochondrial field in general, the methods are still coming together. So it's really important to use good methodology when you're looking to see, especially persistence. So we always use methods that are either looking at genetically encoded proteins and not using any like Mitochek or green or any dyes, because dyes tend to stick to any membranes. We identified a protein that's specific to placenta-derived mitochondria, so we can track whether it went into the hematopoietic stem cells using specific protein staining and using the single nucleotide polymorphism. So we use a lot of different methodologies to look to see whether there's persistence of the allogeneic exogenous mitochondria. And also important in all of these cases is to make sure you're only looking at internalized mitochondria. So we don't want to see any leftover mitochondria that are swimming around in any of these conditions and falsely quantitate them as mitochondrial transfer. So using really stringent methodologies, we looked at the erythroid differentiation assays, we looked in the different hematopoietic tissues in the in vivo studies, and we later looked in human, and we don't see persistence long-term of the exogenous mitochondria. That might explain why it's not really critical to do any haplogroup matching between the donor and the recipient mitochondria. And so we're starting to ask ourselves what the mechanism is, and this is definitely a work in progress. What we think is going on is that there's a mitochondrial recycling. So as maybe I could share this preliminary result, if we look at patient cells before at baseline and after we create the drug product from them, and we look at their membrane potential, we know that membrane potential can have a profound influence on the activity of hematopoietic stem cells. And what we see is that there's a shift to cells with a higher membrane potential. And that shift is also correlated with a higher mitochondrial copy number. And it's also mitophagy dependent. So if you bring in a mitophagy inhibitor, you lose that rise in copy number and you lose the shift in membrane potential. And what we think is going on is mitochondria are coming into the cell. They're causing a shift that leads to mitophagy and it leads to mitobiogenesis. And so it shifts the mitochondrial balance in the cell. It's kind of causing this recycling. Um, and then as time goes on, the exogenous mitochondria are lost, but the effect of the mitochondrial augmentation remains. And this is very similar to a relatively recent published study with a similarly proposed mechanism of action. They're both very consistent, where mitophagy occurs, and that's what's mediating the improvement. So after all that, we wanted to make absolutely sure that we feel safe going into myelodysplastic syndrome patients. And we did a collaboration with Memorial Sloan Kettering with Omar Abdel Wahab. And what we did there was take a mouse model of an NHD13. So this is a fusion gene that causes a murine phenotype very similar to the human disease. They have cytopenias, they progress to AML. And what we did was we took cells from these animals or from wild type controls. We augmented them with traceable mitochondria from a different variant of mouse. So it's an NZB mouse. And then we administer these cells into a wild type mouse that has been irradiated. Okay, so this wild type mouse now got either um, MDS cells or wild type cells that were augmented or not augmented. And what we were surprised and excited to see was that the mitochondrial augmentation actually suppressed the leukemia onset in this mouse model. So what you see here is this is what happens to the mouse. It dies a little of after a month. This was 10 different mice. Whereas if you did mitochondrial augmentation, we got a significant suppression of the leukemia. They did also succumb to leukemia in the end, but it was much later. And nothing happened to the wild type one way or another. So, which is also important from a safety perspective, to do no harm. So if you do mitochondrial augmentation on a wild type animal, nothing bad happens. Um, and here too, there was no long-term persistence of the exogenous mitochondrial DNA. So again, it doesn't persist. The allogeneic placenta-derived mitochondria doesn't persist over time, but the effect is extremely durable. And so once we saw this, in addition with the data on erythroid differentiation improvement and in vivo improvement, we decided we feel comfortable going into a clinical trial in myelodysplastic syndrome. And I'm just going to share preliminary results from one patient. He was our first responder patient, so we're really excited by him. This was a 69-year-old male. He had hypercellular bone marrow, a lot of the symptoms very common in MDS patients. And he'd actually received every possible therapy that's been approved, so erythropoietin and luspatercept, and hadn't responded to any of them. We treated him with MNV201, so this is our second generation product. First, we were excited. There is no immune response to allogeneic mitochondria, and that's always important because it's always a question we keep getting. And we followed him. There's no negative alterations in the bone marrow. But the efficacy was what was really exciting. So if you look at the top right, the blue bars are the number of transfusions this patient received every month. So you could see it gets usually 3 to 4 transfusions. And the red is his hemoglobin. So in 2023, he got erythropoietin, didn't really respond to that. The hemoglobin was still low. And then he got luspatercept, didn't really respond to that. The hemoglobin was still low. And here in August, he received our treatment. And you can see that after 2 months, he became completely transfusion independent. And the hemoglobin rose to nearly normal levels and it's still staying there. So it's been a year now. In parallel, one of those biomarkers, the GDF15 that I showed you earlier, dropped to normal. And actually, the bone marrow showed that he actually transitioned from being a low risk MDS patient to a very low risk MDS patient. And I'll circle back to what maybe the first slide that I showed you with the Pearson syndrome patient. So the very first Pearson syndrome patient that we treated, we saw improvement in his kidney function, which has surprised us. So this patient actually also had renal function that improved. So he had his creatinine and his bun to urea ratio returned to normal after treatment. His nephrologist was ecstatic and surprised and said that never happened in his experience. So we're really excited by that. And again, I think we're still not 100% sure how this treatment in hematopoietic stem cell mitochondrial function is translating into improved renal function, but it's something we've seen in more than a couple of patients. So we're really curious to see where that leads us. Um, so just to summarize and leave as much time for questions as possible, um, the current understanding of the mechanism of action that is still undergoing active study. And I think that's one of the things that kind of you have to balance in a company pushing forward the clinic versus the amount of effort that you can put into understanding mechanism of action and to doing preclinical studies. Because in the end, our goal is to treat patients and to make sure we treat them well. So we saw is that the level of augmentation in the hematopoietic stem cells is about 30 to 50% and up to 30% exogenous mitochondrial DNA enter per cell. Um, we don't see persistence in anywhere in vitro and in vivo in human studies, despite the fact that we see durable effect. We did actually see that if you stall the cell cycle, you do see persistence. So it is something, it's not, and that it's not the mitochondria that are coming in are not going into the lysosome. So they're not undergoing active degradation, but they are somehow slowly getting swamped out over time. So the exogenous mitochondrial DNA is not replicating. We don't see the need for haplogroup matching in this instance. So as long as there's no persistence of exogenous mitochondrial DNA, I don't think haplogroup matching is necessary. We didn't see any difference from a safety perspective, and we didn't see any difference from an efficacy perspective. We saw the effect of mitochondrial augmentation both in vitro and in vivo, as I showed you, on the different lineages and engraftment and reconstitution. And we didn't see any immune reaction to allogeneic mitochondria. And I didn't mention it, but we've already redosed several patients. So there are patients that have now received more than one dose of allogeneic mitochondria. And even after that second dose, there's no immune response. And I think that's important because anybody coming from a gene therapy field knows that you can only give a gene therapy once, right? So if you got AAV once, you can't get it again. And so that was a really big question from our point of view with respect to allogeneic mitochondria. So ongoing work in progress, understanding mechanism of action, but we're really excited to be in 2 clinical trials in parallel and to continue to see that we're helping patients. And I'm really happy to want to thank all of the, academic and medical institutions that have been working with us. And I'm happy to take any questions.
That was
Thank
so
you so
great.
much. Yeah, I think there's like I don't think there's a single company that's as inspiring to me personally compared to what you do. I can start us on with one question. I have like seven written, so um I'll start with one. Huh. Which one should I pick? Um hmm. Oh, from a clinical standpoint, why does the FDA not like IPMDS?
So when you look at the IPMDS, and there's 3 different parts of that score. There's functional scores, there's patient-reported outcomes, or actually in this case, it's caregiver-reported outcomes. Some of them tend to be squishy. We actually had a couple of conversations with the FDA. So some of them are measurements, how well do you see your visual measurement or your height or your weight? That's very objective. Others are, how does your child feel? How is he eating? Is he vomiting? Those types of questions. And they can be, they're not necessarily 100% objective, right? If I vomited yesterday, but when I'm testing at baseline, but at 6 months, the patient hasn't vomited for a week or two, parents might be forgetful. So I understand why the agency is reluctant to take that as a purely objective endpoint. We have been looking at the more objective questions and considering doing a partial IPMDS with just the objective questions or the measurements. Yeah.
I have a question, unless you want to continue.
No, I'm not gonna do the seven.
Okay, we're gonna continue. So I have, I was just wondering because there is no persistence, right, of this mitochondria. Was experiment when the donor and the recipient was the same? Was such an experiment performed, and then just, would that be any different than?
So the only experiment where you can really do the donor and the recipient are the same are, for example, the first product, right, where the mother is exactly the same haplogroup as the patient. But then the challenge is you can't really track it. So I can say that we're seeing similar efficacy between the first generation product and the second generation product, and that's one thing. And with respect to all of the other experiments, it was we were we were limited by the whatever hematopoietic stem cells you received and whatever we had in our clinical bank. But we had situations where the match was two single nucleotide variants or three or five single nucleotide variants, and we had situations where there were 30 or 60 or 100 variants between. And we weren't seeing any difference. So we weren't seeing that it was more efficacious if the batch was closer.
So when you mentioned that you cannot track them, so you cannot somehow tag them with, adding something to the cell.
So you can't tag the mitochondria, right? Because there's no way to genetically engineer the mitochondria with some kind of GFP tag or something. When we were doing the experiments with the dendra tag, you lose the signal over time, right? You're limited by um, the half-life of the protein that's getting transferred. And when we're doing it from a clinical perspective, we're not going to put in some GFP or something or into the allogeneic mitochondria. Um, yeah, so I think that's a limitation. I'm happy if anybody has ideas. I know there's a mitotracer now that's out that you can, it'll label cells that receive mitochondrial transfer at any stage in the past. So we are working with Omar Abdul Wahab at MSK to kind of do some of this mitochondrial augmentation technology work with that mitotracer and hopefully be able to answer some of these questions more definitively. Yeah.
Thank you.
Is there any question from the audience? Maybe. Okay, then I will go with my question. First of all, congratulations. It's very inspiring to see your results. I myself work on mitochondrial transfer, and I see how hard it is to get things working in this system that's relatively not studied. So I'm really impressed to see how far you went. And my question is about the immunity, actually. You mentioned several times that you didn't observe any immune reaction against allogeneic mitochondria. So I was wondering why. And also in the case of mitochondria from the mother, because I mean, the mitochondrial DNA encodes only for a few proteins, but all the rest of the proteins decorating the membrane, um, I mean, I would have expected them to cause to some extent an immune reaction. So I'm very curious to hear about
Yeah,
this.
I know. So two reasons. The first reason I think is one of the main rationales that we went with ex vivo mitochondrial augmentation. So there's a lot of kind of work in the past couple of years on direct mitochondrial administration, so mitochondrial transplantation as a clinical product. So people are harvesting mitochondria from a variety of sources and injecting usually into the bloodstream. Um, One of the things that we first thought when we decided to do an ex vivo mitochondrial transplantation was exactly that is we wanted full control over the process when it's outside of the patient's body. And if your mitochondria are going into your hematopoietic stem cells and they're now completely enclosed, and then I wash the hematopoietic stem cells from any residual non-internalized mitochondria and now reinfuse them, what the patient's body is actually seeing is hematopoietic stem cells, right? There's no released mitochondria and released mitochondrial DNA. So that's one aspect of it. The other aspect is part of the quality control is we're making sure that the mitochondria are not ruptured. Sorry. So there's no spilled mitochondrial DNA. We had quite a few discussions with the FDA before going into the MDS clinical trial. So both of the trials are happening in Israel, but they're under FDA IND. And we had asked the agency in advance to approve redosing. And what they said, they were concerned about the potential immunological responses. And so we said, well, instead of just giving us the clear to go on all redosing, we'll come to them on a patient by patient basis with all the data. So in all of our patients, we're looking at anti-mitochondrial antibodies and we're measuring whether we see them at all. And we're not seeing any of them. I think there's a difference in the literature between theoretical, and I can have this conversation with you, Lorenza, afterwards for hours, but there's a lot of theoretical papers that can show immune response, and all the damps and what have you to mitochondrial DNA. And then there's the reality in patients, and the findings don't always bridge from. And I've had this discussion with half a dozen immunologists and mitochondrial experts. We're not the only ones seeing that actually mitochondria do not cause harm. So there's the paper from John Brestoff in the Lee syndrome model from about a year ago. And he actually sees, even with, and this is injecting direct mitochondrial into the bloodstream, improved survival. So I don't know that all of the preclinical studies that look at immunology of mitochondria fully translate into the human.
Thank you. That's very interesting. I would have expected cells containing exogenous mitochondria to expose like some peptides from these mitochondria that
Neoantigen.
don't match the endogenous peptides and the two. So the liver, like the cells receiving the exogenous mitochondria don't have any differential survival in the patient.
No, and I showed you the in vivo studies where you're actually seeing a much higher level of engraftment, so that kind of would negate that. Yeah. Yeah. But and I know there's studies showing that that mitochondrial epitopes are expressed by MHC. So, that's where we weren't sure which way it was going to turn out. But at least, and I always say, at least in hematopoietic stem cells, at least by the process that we're doing, we're not seeing any immunogenicity. Yeah.
That's great. Thank you.
Yeah.
I'm wondering if there's like a different tolerance for like immune reaction for mitochondrial peptides. If maybe in, early development of the adaptive immune system, you have some type of variation in mitochondrial DNA that would allow for that. I'm not sure, but it would be interesting. Like the opposite of the original antigenic sin. It would be interesting.
Yeah, for sure.
I see something in the chat.
Yeah. So someone asked Jordan if Minovia might explore LHON, so Leber hereditary optic neuropathy. It causes blindness later in life, and they said that they have a family member that was recently diagnosed. So I think the eye is unique, right? It's our treatment is IV. And LHON is a nuclear mutation, so presumably anything we do, even if we were able to go into the eye and into the specific eye cells that are dysfunctional in LHON, we're not going to treat the underlying cause of the nuclear mutation. So I'm not sure that MAT might be the best fit. I would suspect that potentially some kind of localized gene therapy might be the way to go in that disease. But I'm happy to discuss it afterwards if you want, Jordan.
And how about other mitochondrial diseases? How generalizable do you think it is to, I mean, both
Yeah.
mtDNA diseases and nucleus encoded ones?
Yeah. So it's a great question. I don't have the answer for entirely. I could tell you how we're thinking about it. When we first thought it was going to be like a gene therapy approach, we were very focused on mitochondrial DNA mutations and deletions. So because we said, we're bringing in a genome, so you can't fix any of the nuclear mutations. We did treat KSS patients under compassionate use, and we did treat like Lee syndrome patients under compassionate use and seen improvements in those. When we moved into MDS, that was a big question because we're not sure that the main mechanism of improved mitochondrial function in hematopoietic stem cells is becoming because of those low-level mitochondrial DNA mutations that I showed you. And the MDS patients also have a lot of genomic mutations, right? So it's a pre-cancerous. They've got mutations in half a dozen genes. So the short answer is I'm not sure. We might be able to have some effect on both mitochondrial encoded and nuclear encoded genes, and it's just a matter of prioritization of testing them all. We always have more that we want to do than we can do from a funding and prioritization perspective. Yeah.
We have a
And I
question.
think it might also be an issue of durability. So we might see a higher durability in some diseases than in others.
I see a question from Andreas in the chat.
How do
Mitochondria.
host cells regulate mitochondrial health? Do they recognize dysfunctional mitochondria? So presumably in a perfect universe, right, when everything is functioning, then and I'm hoping I'm answering your question, but if not, then please elaborate. Then mitophagy would work properly and the host cells would recognize, some mitochondria as being functional and some as being dysfunctional and would get rid of the dysfunctional ones. Obviously, in the cases of all of these diseases, that's not working properly. And I don't think anybody knows for sure exactly why. So why does Pearson's syndrome patient cells not get rid of those dysfunctional mitochondria? I suspect that in some cases it might be additional underlying causes. So potentially some of the mitochondrial nuclear genes are just suboptimal and the whole mitophagy pathway is suboptimal. I also suspect that because the mitochondria are dysfunctional, there's not enough ATP to do all of those energy related processes such as mitophagy. So, this is like a chicken and egg type of thing. But in general, in a normal host cell, they're supposed to recognize dysfunctional mitochondria and get rid of them. That's obviously not happening in these diseases.
Okay, thank you. Yeah, I'm just curious, like from the perspective of thinking about the mitochondria host cell relationship as a symbiosis, um, like if there are patterns on a genomic level of like host genetic variation that correspond with different levels of mitochondrial genetic variation that is linked to dysfunction within certain contexts. But yeah, I mean that's like I think you yeah, I think that's been the question. I know very little about mitochondria, so I really appreciate the presentation.
Yeah. I think there's so much to be still left to be understood with respect to the crosstalk between the mitochondria and the genome, philosophically thinking about it as an endosymbiont. Um, and both are undergoing kind of simultaneous evolution. There have been studies at a population level saying, okay, if we take situations of Europeans going into, I don't know, South America, do you see different levels of is there a problem matching some mitochondria with some nuclear backgrounds? And for the majority of things still work. There are some outliers. Um, but I do think that there are mitonuclear matches, which will give you better mitochondrial health and poorer mitochondrial health. Yeah.
Um, yeah, if you allow me, just one more question. It's kind of related. Are there is mitochondrial dysfunction more or less common in different tissues within a single individual?
Yeah. So in this particular disease or in general? I mean, in general,
In
right?
general,
In general, the diseases that tend to have more mitochondrial dysfunction are just the ones that need mitochondria more. So the brain, the muscle, the kidney, the mitochondria intensive ones. The other thing that's interesting is, so for example, in Pearson syndrome patients, the patients start all with anemia and with a high level of heteroplasmy. And it's known, and nobody really knows why, that specifically the heteroplasmy level reduces over time in the blood and only in the blood. So it doesn't fix itself in the brain or in the muscle or what have you,
hmm.
but it does fix itself to some extent. I suspect it's an issue of competition. So hematopoietic stem cells with lower levels of heteroplasmy are able to survive more and make more downstream progeny and so forth. So those patients that do survive the first couple of years, they have a reduction in heteroplasmy. And um, I think it has to do with proliferation. So the proliferative tissues are able to kind of fix mitochondrial dysfunction to some extent over time, whereas the less proliferative tissues are not.
Hmm.
But I think mitochondrial biology, there's a couple of fields there, right? The immunology and the mitochondrial, they probably are two of the craziest fields, maybe with neurology added in there for the fun of it. Um,
Yeah, yeah. I mean, and there's a whole ecology too with the transfer of mitochondria between different cells, and
yeah,
with variation of mitochondria and then different regulatory backgrounds within different tissue types. when you bring in like competition and fitness into the mix, then you get all kinds of different like pleiotropic effects. I mean, that's just bananas. Wow.
Yeah.
Thank you very much.
Can I ask one last question, small one, and then we can finish? Or does anyone else have one? Okay, I'll ask one last small question. Is there any type of dose dependence that you can see with the mitochondria that you provide?
We did, so what we have a specific mitochondria to cell ratio that we do in our process. We did a dose escalation study in the early days to see what is the minimum amount of mitochondria that you need to put in a cell to get the maximum mitochondria in and the maximum efficiency. Yeah. At some stage, you're just at diminishing returns. You've got tons of mitochondria, and what's limiting is something in the cell biology. So specifically, hematopoietic stem and progenitor cells are almost all nucleus. There's very little room for mitochondria if you look at a microscopic slide of those. So I think what's limiting there is just the biology of that particular cell type more than anything else. But there is a dose limitation at some stage. Yeah.
Thank you so, so
Can,
much.
can
I think
sorry, I have just another small question. If
I'm
because
here.
so I don't work with mammalian cells, but we work with some diatoms, and we've been trying to capture, and we actually capture whole genomes of mitochondria. They are bigger. They're anywhere from like 40 to 70 kb, so they are bigger than humans. And we try to develop technology for whole genome replacement in mitochondria. Is that technology will be also useful for mammalian cells? If
If
there
you
was,
have the technology and it translates, it would be amazing. Yeah.
so
So you're swapping out the you said diatom, right?
diatoms, so we can capture them now in host organisms, whole genomes.
Yeah.
We still have to figure out how to put them exactly in. So that is the delivery is, but I'm just again, it's all diatom, but I think with human cells that would be kind of potentially. I see some advantages, obviously, with a smaller genome and a couple of things, but yeah, I just wonder how. I mean, I guess it would be useful, right, to be able to do that.
Yeah, for sure. So there's a call for proposals from ARIA, the UK, to do precision mitochondria, to try to do genomic editing of mitochondria. It's something that's like nobody's been able to do. You can't really bring in DNA or RNA into mitochondria. Um, and so you could bring in proteins with a mitochondrial targeting sequence. But if you could genetically engineer mitochondria, that would be amazing. Yeah.
Bogomil, if you want, we can try. We're trying to do a study together, but I didn't realize you're interested in this. We should talk later. You should try what we're building here.
Sure.
Email me if you would like. I can send you stuff. Okay, let's finish now because we're already over time. But thank you so much for starting us up. Yeah, please share the seminar with anyone who you think would be interested. This was an awesome start for the next semester or year of symbiogenesis seminars, and we're excited to see you all in 2 weeks.
Thank you.
Have a great year.